All right, everybody, good morning. We're talking about the sponges or the porifera. Hopefully you remember that por means to have holes and fer means to bear. So porifera means it's going to have holes, and if you look at these sponges, you can see that they have holes all over their surfaces. Remember, in kingdom Animalia, we're talking about phylum Porifera. The next phylum we're going to talk about are going to be the cnidarians, which are going to be corals, but in this test we're talking about phylum Porifera, which are the sponges. We have this Hexantenellida, the Sclerospongiae, Demospongiae, and the Calcarea. Those are the different classes. Remember, spicules are the support structure. Some are going to be made out of glass, and some are going to be made out of calcium carbonate. Spicules are like their skeleton. Spongin. Spongin is going to be found in the Demospongiae and the Sclerospongiae. It is going to be this fibrous collagen substance that really soaks in water, and basically it is the skeleton of a dead sponge, usually the Demospongiae. There's the Demospongiae. Remember, people have been using these for a long time, so they are called the people sponge, and the class name Demospongiae reflects that. And remember, they are the ones that make the spongin. The Calcarea, of course, they have calcium carbonate spicules. Calc is in their name, and calcium carbonate is what their spicules are made of. The Hexantellidae, remember these guys have 6 pointed spicules right here, and SiO2 is going to mean glass. And don't forget this hex in the name. This hex means 6, and they have the 6 pointed spicules. Okay, we're talking about asexual reproduction. Sponges asexually reproduce in about 3 different ways. The first one is going to be a gemmule. If you look here, here's a little close-up of some of these. You can see they're surrounded by spicules, and if you remember, if it's going to be asexual reproduction, it's going to have some archaeocytes inside of it, right? Some of these archaeocytes right here, those are the ones that are totipotent, and they can become any type of cell. Here's what these actually look like to the naked eye. We're talking about gemmules, asexual reproduction. The next form of asexual reproduction is simply budding. Remember, when a sponge is growing and it's nice and healthy, it's going to have little babies like these little guys right down here kind of pop up around the edges. They're going to be clones of the adult, and remember, this is going to be through mitosis, right? It is not sexual division. This is just asexual growth called budding. The last form of asexual reproduction that these sponges have is going to be called fragmentation. Remember, it's still mitosis, and basically what we're going to see is a little fragment like this is going to break off and grow. Remember, a fragment is a tiny little piece, and this is called fragmentation. Remember, they do reproduce sexually also by releasing what we call a haploid egg. Remember, it has 1 set of chromosomes. The haploid sperm has another set of chromosomes, and when they come together, they make a zygote that has 2 sets, one from mom, one from dad, in case one isn't working. And remember, the sexual reproduction, this is going through meiosis to make this egg and this sperm. All right, we have the... Okay, I'm back. Sorry, that was my wife calling. All right, guys, so now we were talking about the choanocytes. What you should remember is that choanocyte means funnel cell, and these little cells right in here that line the inside of the spongiceal on this particular type of sponge are going to be the choanocytes. Remember, they make the current, and they're going to capture the food. The choanocytes. Next, we're going to have the penicocytes. Penicocytes are going to be the skin cells. We have shaded these in quite a few times, guys. Please do not miss the penicocytes. They are going to be the skin cells, the ones that cover the outside surface. Next, we're going to have the ostia. Remember, ostia is where the water is going to be going in. These small little holes right over here are going to be where water is going into the sponge, and they are called ostia. After that, we're going to have the osculum. Remember, everybody, the osculum right here is where the water leaves. Water is going to head up this direction out of the osculum where the water leaves. We have the archaeocytes, everybody. Now, they're a little bit hard to color, but we're talking about these little amoeboid-looking things that are in the mesenchyme right in here, right there in this middle area. These things are totipotent, which means they can repair a damaged sponge, and they're also going to pass food around to the other cells since they don't have a digestive system. Spicules, remember everybody, spicules are going to be the support structure. We've seen them a few times. Here's some stabby little spicules sticking out here and here and here. You can see them in the mesenchyme. They are a support structure and sometimes also useful in defense because some animals do not want a mouthful of glass. Remember, we're going to have what's called the asconoid sponge, the syconoid sponge, and the leuconoid sponge. What you need to remember about the asconoid is where this red is right here. This is where the flagella are. They're in the spongiceal in the asconoid sponge. Now, in the syconoid sponge, it's folded on itself, and the flagella are actually going to be inside walls, and these are usually going to be shaped like tubes. In the leuconoid system, we're going to see that they have all of these numerous flagellated chambers all over the place. It is very efficient and feeding, and the Demospongiae are going to have this sort of leuconoid canal system, so it is the most popular. All right, and then lastly, guys, exocytosis and endocytosis. Everybody should remember that exo means out. So when something is moving out of the cell, it's going to be called exocytosis. And when things are moving into the cell, it's going to be called endocytosis. That's most of the stuff you need on this test. Good luck, everybody.